Intermittent fasting, IF, a dietary strategy that cycles between periods of eating and fasting, has gained significant attention not only for its potential in weight management and metabolic health, but also for its promising effects on the aging process, particularly in relation to telomere dynamics. Unlike continuous caloric restriction, which reduces daily calorie intake, intermittent fasting relies on periods of voluntary abstention from food, ranging from hours to full days, without necessarily reducing total caloric intake. What makes it especially intriguing is the growing body of evidence from both animal models and human trials that suggests it may help preserve telomere length and enhance cellular longevity through mechanisms rooted in evolutionary biology and stress adaptation. One of the key processes stimulated by intermittent fasting is autophagy, a critical cellular cleanup system that becomes activated in the absence of nutrients. During fasting, cells shift from a growth and storage mode into a maintenance and repair state. Damaged proteins and organelles, especially dysfunctional mitochondria, are cycled and removed, which reduces oxidative stress and enhances mitochondrial efficiency. Because oxidative stress is a major driver of telomere shortening, the activation of autophagy under fasting conditions is believed to help protect telomeric DNA from unnecessary damage. Moreover, it improves mitochondrial health not only by encouraging the removal of damaged mitochondria, but also by promoting the biogenesis of new, more efficient ones. This dual effect contributes to a less inflammatory and more metabolically resilient internal environment, conditions that are highly favorable for maintaining telomere integrity over time. Fasting also triggers a mild hormetic stress response, in which short-term, low-level stress leads to long-term adaptive benefits. This is a powerful concept in the biology of aging, the idea that challenging cells in controlled ways actually strengthens their defenses and resilience. In the context of telomeres, this hormesis appears to enhance the expression of protective genes and activate pathways that stabilize chromosomal ends. Studies in rodents have demonstrated that intermittent fasting can preserve telomere length even in the absence of overall calorie reduction, and some animal models show extended lifespan and delayed onset of age-related diseases under intermittent fasting protocols. In human research, while long-term data are still emerging, early findings are promising. Individuals who follow regular intermittent fasting regimens, such as the 16:8 method, alternate day fasting, or 5:2 weekly cycles, often exhibit reduced markers of oxidative stress, improved insulin sensitivity, and lowered systemic inflammation. These changes mirror the internal conditions seen in slower aging cells and are thought to indirectly support telomere stability. Additionally, fasting appears to enhance metabolic flexibility, allowing the body to switch efficiently between fuel sources, which contributes to overall energy balance and reduces metabolic strain that could otherwise promote telomere shortening. What makes intermittent fasting particularly attractive is its accessibility and adaptability. It does not require specialized foods, supplements, or costly interventions. It aligns naturally with human physiology, reflecting ancestral eating patterns shaped by periods of feast and famine. Moreover, it can be tailored to fit individual lifestyles and preferences, from daily time-restricted eating windows to more prolonged fasts practiced less frequently. For many, this flexibility makes it a more sustainable and psychologically manageable approach than strict, continuous caloric restriction. Intermittent fasting also pairs well with other health-promoting behaviors, including exercise, nutrient-dense eating, and stress management practices, all of which can amplify its benefits. Its potential synergy with other anti-aging interventions, such as telomerase activation, caloric moderation, or even targeted supplementation, opens exciting avenues for personalized longevity strategies grounded in evidence-based science. While further longitudinal studies are needed to directly confirm IF's long-term effects on telomere length in diverse human populations, the mechanistic data and early human results already support its use as a lifestyle tool with real potential to slow biological aging. Far from being just another dietary trend, intermittent fasting stands out as a scientifically sound, evolutionarily informed, and practically achievable approach to cellular health and vitality. Its influence on telomere maintenance underscores a broader truth: that strategic timing, not just content, of food intake can have profound implications for how well and how long we live.